Okay, our next speaker is Katherine Coyte from Manchester.
All right, cool. Everyone's settled? Yeah. Hi, I'm Katherine Coyte. I'm a group leader at the University of Manchester. And as promised, I'm going to talk today about some work that we've been doing thinking about early life microbiome assembly. But, you know, the title of this workshop and everything is sort of, what is it? Like open challenges and current approaches, or maybe current challenges and open approaches, I don't know. But I thought, while I'm going to tell you about a specific sort of scientific story, I might also use this as an opportunity to talk about at least what I think are, in my personal opinion, some of still the big challenges that we face when studying microbial communities. So first, some introduction. When I talk about microbiomes through this talk, I'm going to be really lazy and use that as a catch-all term specifically for the human gut microbiome. So that needs no introduction. We all harbor this sort of diverse community of microbes within our guts that's really important for our health and well-being. But what I think is way less appreciated is that we are not born with this microbial community, right? So in utero, we are sort of virtually, if not literally, sterile. And what that means, right, is that the moment that we're born, we have to somehow each build our own unique microbial community. And, indeed, this is what we do. So if you look over time, what we see in infants is that we see this sort of gradual development of the microbial community within an infant's gut as they age. And this process isn't instantaneous. In fact, it's really not until you're about sort of 2 or 3 years old that your gut microbiome kind of resembles roughly that of a healthy adult, kind of assuming nothing goes wrong. And one of the things that I think is really remarkable about this process is that it's incredibly predictable. And what I mean by that is that if we look at sort of infants at specific points in time, we see that there tend to be certain taxa that colonize the infant gut at certain time points. So we have some kind of, pioneer species that get in there almost immediately, and then we see other taxa that don't establish within the infant gut until really sort of weeks, even months after they've been born. And this predictable developmental process itself seems important for health. So the wrong microbe getting into the infant gut at the wrong point in time can have really profound impacts on infant health. Yet, despite this sort of clear importance and this interesting scientific topic, we still really do not understand what shapes this developmental process. So what is it that makes our microbiome develop so predictably? And we know some kind of coarse-grained things. So we know in full-term infants, for example, whether you were born vaginally or via C-section plays a massive role in shaping those initial colonizers. And we know kind of further down the line, the transition from milk or formula onto solid food, huge shift in microbiome composition. But beyond these bookends, right, these very coarse-grained sort of aspects of microbiome dynamics, we really do not know what is shaping this developmental process. And so a lot of what I work on and what now my lab works on is we're really trying to understand what determines how a microbiome assembles over time. And that's what I'm going to talk to you guys about today. But in particular, for the vast majority of this talk, I'm going to tell you one specific scientific story where we've been trying to understand the specific drivers of microbiome assembly within one particular cohort of infants, so one particular set of infants at a hospital in Boston. And then if I get time, and I'm not sure if I'll get time, but if I do get time, at the very end, I'm going to talk more generally about, how can we use this work to think about more general rules that we think might shape microbiome assembly in any infant or even in any mammal, for example. So that's a grand idea. So first things first, how can we identify specific drivers of microbiome assembly? When we started thinking about this question, years ago now, one of the first things that was obvious to me is that if we want to understand microbiome assembly, the first thing we need to do is we need to describe it, right? We need a lovely data set that tells us how the microbiome is developing within, say, a certain set of infants. And when I was saying this is what we need, everyone sort of turned around and said, well, at this point in time, there are so many data sets, right? So if you go onto PubMed or whatever and you type in even infant microbiome, you will see, tens, maybe even 100 now, different data sets, amplicon sequencing, microbiome dynamics over time, right? But in my opinion, and this is my first sort of, pitch for current challenges in microbiome dynamics, in my opinion, at least in the human microbiome world, most data sets are still plagued by a few real key sort of humps, key issues with the data sets. And the first one is this matter of timing, right? And this wasn't so much the case in these beautiful anaerobic digesters that we saw this morning, but if you think about most microbiome data sets, we are typically looking at someone's microbiome, maybe every few months, perhaps every few weeks if we're lucky, if we're looking at dynamics over time. But of course, if you think about most microbes, they're rapidly growing within the gut. If we want to understand, if we want to really understand what's going on, how communities are changing, we need to be sampling every week, really every day, right? If we want to actually see how a community is changing over time. So that's kind of issue number one. The second issue, and I think is it Amelia talked about this so nicely in her talk, right? Which is that overwhelmingly still when we study microbial communities, we are super bacteria-centric, right? So we do our 16S amplicon sequencing, we chart all of the bacteria that are there, and we tend to completely ignore all of the other organisms that might be there and might be playing a really important role. So, fungi, archaea, viruses, all these other microbial kingdoms that might be present, right? And the final one, and this is my singular obsession, right? And I think I've used this slide for a while, and it used to be that people would say, I don't know what you're talking about. Now I think this is more appreciated. So really, when we are studying microbial communities, we tend to be looking at relative abundances, right? So we look at the proportion of a particular microbiome that is made up of a particular taxa, the different percentages, right? And this is great, this tells us a lot, but it really confounds our ability to understand microbiome dynamics, right? And just sort of trivially, it's a bit small here, I guess, but my toy example, imagine you're looking at a person and you see a particular, let's say, species increasing over time within this person's gut, right? We do not know. Could be that this microbe is growing like wild, it's proliferating, it's some sort of horrible food poisoning bug, it's growing like wild within their guts, Or it could be that perhaps my patient, maybe they've taken an antibiotic, most bugs are dying within this person's gut, and actually this microbe here, it's just doing what it's always been doing, but it happens to be resistant to that antibiotic. So it looks like it's growing within this person's gut, but actually it's really not, right? And our inability to disentangle these different processes really confounds our ability to understand dynamics. So what we did a while ago now was to say, well, let's just build our own data set, but one that overcomes each of these challenges. And so in particular, working with this absolutely phenomenal person. So this is Cami Martin. She's a clinician scientist. She was in Boston, now in New York. We assembled a cohort of around 200 preterm infants whose microbiomes we sampled as frequently as we possibly could. So in some cases, every single day for their first 6 weeks of life. And I want to emphasize here, we are looking at preterm infants. So this is infants born before their due date. This is a very particular cohort of infants. They have their own unique aspects of sort of life. The advantage of studying preterm infants is that they are probably the most sort of vulnerable and carefully cared for patients that you will find in a hospital. And what that means is that we have incredibly fine scale detail on every single antibiotic, every clinical intervention, every single thing that happens to each of these infants we know about, we have a record of. So they are a great study system, actually. And what we did then, and this is really the work of this guy, Chitong Rao, who at the time was a postdoc in Seth Rakoff-Nahum's lab, is develop a method to quantify sort of the absolute abundances of not just bacteria, but also fungi and archaea within each of these samples. And in particular, what we use is this multi-kingdom sort of cell spiking-based approach. And we can talk about the nuances of what it means to really have the absolute abundances, but really what this enables us to say is genuinely from one time point to the next, are microbes growing or dying within a person? So this is the idea, this is what we did. What did this tell us? So the first thing that we see is within our preterm infants, indeed, their bacterial communities are sort of develop in an incredibly predictable way. So this is a standard ordination plot of the bacterial communities within all of our infants. What you'll see is that their gut microbiome tends to cluster into one of really just 4 distinct states, characterized by the domination of one particular microbe. So this is Staph here in yellow, Klebsiella in blue, Enterococcus in green, and Escherichia in red. And if you look here, instead, if we color these plots by this sort of dots by infant age, we see there's this sort of age-based transition. So almost all of our infants start out life dominated by Staph, and as they age, their microbiome transitions into one of these other 4 distinct states. So this very characteristic sort of stepwise progression of the gut microbiome. What we also see that we didn't know before is that you also have a sort of predictable development just in terms of total bacterial load. So we see this incredibly rapid initial colonization of the infant gut happening really in the first sort of week to 10 days of life, and then this slower gradual sort of increase in total bacterial load afterwards. And so I want to really highlight this point here, right? We're seeing rapid dynamics happening within a week. So really emphasizing this importance of capturing the time scale that's relevant to your system. So that's the bacteria, that's what we see. If we look at the fungal communities, actually, we see very different dynamics. So we see, similarly, we tend to see the infant gut dominated by one of just a handful of different fungal genera. But what we don't see, we can't see this by eye, and neither can we pull it out sort of statistically, is we don't see any map between fungal community composition or total fungal load and infant age. So instead, what it seems like is that most of our infants harbor a sort of a low abundance, low diversity fungal community, but we have these sporadic and seemingly completely unpredictable blooms in total fungal load that happen every now and again. So this is neat. I think it's neat, right? We have these sort of, 2 very different dynamics occurring within these 2 different microbial kingdoms within the preterm gut. What's cool though, again, I think it's cool. What's cool is that while we see very different overall dynamics, actually we can pull out apparently a relationship between these 2 microbial kingdoms. And in particular, what this is if we fit just a standard linear model on total bacterial load, we pull out this weak but very robust sort of inverse correlation between total bacterial load and total fungal load within the infant gut. And so this is, you look at this and you think, are you seeing some sort of competition, right? Some sort of competition of the ability to colonize an infant's gut between these 2 microbial kingdoms. But the thing is, right, from a plot like this, you cannot say, right? We do not, this is purely a correlational analysis. And this is my second kind of, in my opinion, big open challenge in microbiome science. If we want to understand how, and I'm really laboring a point here, everyone knows this, right? But if we want to understand how any particular sort of microbial taxa behaves within the gut, we need to somehow pull out, well, okay, what's the effect of the host, right? It could be that these infants, they've just been born, they're preterm, they're actually, their immune system, for example, is rapidly developing over time. So there might be effects of the host. These kids are getting all sorts of antibiotics. Each kid is getting a completely different cocktail of different antibiotics, almost certainly going to be affecting the growth of each individual microbe within the gut. But then, as we've heard so much about, right, there's also going to be all of these microbe-microbe interactions. We've got this cross-feeding, we've got competition, we've got, I really love these examples of the sort of exploitative predator-prey interactions that we've seen so far, right? We've got all of these different microbial interactions that could also be driving the dynamics that we see, right? And, this is this thing, right? How we've got all of these things going on, we've got these correlational relationships, and how do we pull out which of these different factors is causatively driving the dynamics that we see? And this is this massive challenge that all of us face, whether we are studying anaerobic digesters, human guts, zebra guts, whatever you're looking at, right? So what do you do in a scenario like this? So what we do actually is really go back to an idea from very classic theoretical ecology. And this is this idea that you can take really any ecosystem, right? And you can model it using a very simple mathematical equation. And I imagine lots of you will recognize this. This is an extended version of the classic Lotka-Volterra sort of community dynamics, first developed for foxes and hares and all sorts of other things a long time ago. But basically, we're just saying, okay, let's assume that each taxa within a community its growth rate from one day to the next is going to be a sort of a function of its own intrinsic growth properties, any interactions that it has with other members of the community, and of course any interactions. So this is the extension that neither Mr. Locke nor Mr. Volterra really thought about, which is any sort of environmental factors as well that might also be driving what's going on. And so what we do is we say, all right, let's assume we can model microbiome dynamics using this equation. Well, then let's just take this equation, let's fit it to our data, right? And in particular, what we do is we fit this model to our data using an incredibly conservative machine learning framework. And what this does is it's basically enabling us to ask, well, can I pull out the cases where the growth of a particular, say one given microbial taxa is really strongly and consistently being affected by the presence of another. So rather than just looking at correlations and total bacterial load now, what we're actually trying to do is pull out the genuine, so for example, microbe-microbe interactions that we think are causatively driving the patterns that we see. And if we can do this, right? If somehow by fitting a model to your data like this, you can magically pull out causative drivers of dynamics, why isn't everybody doing this? Well, really it comes down to the fact that, oh, I've made it tiny here. Let's make it big, right? For this to work, you need to know this. So you need to know, is a microbe growing or dying, for example, or at least increasing or decreasing in abundance from one day to the next, right? And in order to do that, you need absolute abundances, right? Relative abundances are not good enough. You do not know whether something is growing or increasing or decreasing. And of course, also you need a relevant time step. It's no good trying to think about, fit these models on data from, even a 2-week window. So this is why this powerful approach really still isn't used that often in microbiome science. So that's the idea. This is what we do. We take these models, fit them to our data. What do we find out? Well, the first thing is that we pull out, oh, it's rendered terribly again. I need to learn that this figure doesn't look nice. So basically, we pull out this really rich network of interconnected interactions, right? And I want to emphasize this is now not a correlate, or at least not a classic, co-abundance network here. What this is a directed network saying this is how we think different microbes are sort of causatively affecting one another within our infant cohort. Where it gets neat, though, is if we look at those key taxa that we saw that tend to dominate the infant gut, because what we see is that they all seem to be interacting with one another. But what's particularly interesting is, hopefully you remember, right, most of our infants start out dominated by staph, and as they age, they develop, and in particular, they often go from staph to Klebsiella domination. Well, what our model is predicting is that this predictable pattern, it's not being driven by just sort of, some environmental factor or whatever, but actually that we have this exploitative interaction, that Staphylococcus is getting in, it's colonizing, and Klebsiella is then taking advantage of it and using it to sort of gain a boost within the infant gut and to better colonize. So we've got this sort of cool, plus minus interaction. What it is, we don't know. The other thing is that we see this sort of web of inhibitory interactions occurring between these dominant bacterial taxa and this one particular fungus, Candida, that we see often in our infants, right? So if you recall, we saw this inverse correlation between total bacterial and total fungal load. Well, what our model is suggesting, at least, right, is that this isn't just some sort of correlative, reflecting, what else is going on in these babies, but rather that genuinely we do have this competition for occupancy of the preterm gut, that fungi and bacteria really are competing to colonize. So this is great. I've whizzed through a lot, right? So I've said, well, we've taken all this data, and now I'm telling you these are the microbe-microbe interactions that are driving the dynamics that we see. Why should you believe me, right? And more seriously, right, this is this issue when you're using any kind of, particularly these gnarly machine learning approaches, right? The predictions that you get out are only going to be as good as, the assumptions that you've made, the model that you've chosen, right? And we really wondered, is any of this real? But the nice thing is we have a bunch of interactions. We can isolate actually all of these taxa. The preterm gut microbiome is phenomenally aerobic at the start. So these are nice, easy to grow bugs. We can isolate them from stool and we can grow taxa either on their own or in co-culture. And we can actually ask, well, do we see any of these interactions that we've predicted to be so important? And what's nice is that the short answer basically is yes, right? So we can go through and, for example, we see staph being really strongly inhibited by Klebsiella over here. We can also see Klebsiella in turn being inhibited by Enterococcus, which we predict. And most importantly over here, we can recapitulate each of these cross-kingdom inhibitory interactions. So it seems like our model is actually working really well, right? That we can go from this messy clinical data to inferring genuinely what's driving dynamics. But maybe some of you are already eagle-eyed enough to have spotted this, right? But there was, disappointingly, one interaction that we cannot validate in our in vitro system. And of course, it's the most important one, right? And that's that we could not see any positive effect of Staphylococcus on Klebsiella. And when we saw this, I was so disappointed, right? I was thinking, oh my God, this is our incredible story. It all hangs together really nicely. And then the most important thing, you can't recapitulate, right? So what could it be? Could be that our model is wrong, right? Could be that there's something else going on that we're not accounting for. But in a last ditch kind of, well, you know, we were talking about this and saying, well, what else could be going on? And of course, we've taken an interaction, we've inferred it from, you know, what's going on in a baby's gut, and then we are trying to recapitulate it in a test tube, right? It's a world apart. So what we asked was just, well, actually, you know, what if we try and redo basically this co-culture experiment, but instead in a context that matters, right? And in particular, what we did is we took mice with defined microbial communities, and we did basically exactly the same experiment. So, you know, either colonizing them just with Klebsiella, just with staph, or the two together, and then looking at dynamics within these mouse guts. And beautifully, really, you know, wonderful, made us really happy, is that suddenly you do see this positive relationship. So you can see that Klebsiella can colonize the mouse gut way faster in the presence of staph than when it's on its own. And actually, poor old staph, as Klebsiella begins to grow, it does really badly. So indeed, we're seeing that sort of exploitative relationship. But we're also really seeing that context matters, right? That kind of, and I think this is, you know, again, a personal opinion point, right? So many of us, including myself, we're trying to build these lovely synthetic communities where we're nicely characterizing how everyone's growing, you know, in vitro, right? And I think this just really does labor the point that we all know is important, but we try and ignore, which is that if you're trying to understand at least what's going on in a host, you cannot forget about the host, right? It is there, it is almost certainly altering the ecology of the communities that we're studying. And you know, we have to recognize that it's there at some point. We have to go and talk to some immunologists if we, you know, much as I hate to do this, for example. So that I whizzed through a big story, right? And so I promised you, first of all, right, that I would talk to you about identifying specific drivers of microbiome dynamics. I've hopefully convinced you that with the right data and my personal obsession with absolute abundance data, right, and combining that with sort of very classic ecological theory, you can actually pull out sort of, you know, factors that are genuinely driving dynamics within complex data. And also on a more biological note, right, this emphasizing that both within and between kingdom microbial interactions really are critical to driving the dynamics that we see. And this is nice, you can go and read about it in this paper, which I think is lovely, you know. There's also a bunch of other stuff that I didn't talk about. But for the last, I think I've got like 3 or 4 minutes, I just wanted to talk really briefly about a final point, right, which is, if you think about actually what we did. So what did we do? We looked at a bunch of babies, we tracked their microbiomes over time, and then we pulled out the interactions that we think are driving the dynamics that we see. This is great, you publish your Nature paper, hooray, everybody loves you, it's great. And you claim to understand what's driving microbiome dynamics, but do we really from this understand what's driving community dynamics more broadly, right? I frankly would say no, right? If you really want to understand what's going on in a system, well, I'd want to ask, well, what happens if my babies are colonized with a different microbe? What happens if Klebsiella comes in first? Would I expect to end up in the same community state, or might my microbiome end up developing very differently? Or, really what I want to do is say, well, what happens if I change how everything's interacting? What happens if I randomize all of these different interactions? Do I, what happens then to the communities that end up in these infants' guts? And of course, right, we cannot do this. It's very tricky. I guess you could, in theory, do this with some nice genetic engineering, but good luck to you if you want to find a neonatologist who's going to let you mess around with, as I said, the most vulnerable patients that you will find within a hospital. Right, and so this really is where, actually I first started as a microbiome person, which is, oops, I've skipped a million slides, is a very different way of thinking about community dynamics, and that's to think about them theoretically. And in particular, this idea of rather than using these models as a way to fit data, instead using them as a way to simulate, basically do the experiments that we cannot do in reality, right? So for example, you can take these lovely Lotka-Volterra equations and you can just simulate, okay, actually, how for a given community, how would I expect it to behave over time? Or thinking about our previous speaker, thinking, how might I expect it to respond to a perturbation? So something like antibiotics. And I'm kind of overrunning a bit, so I'll be really quick here. There's many different things that we can ask with these more mathematical approaches. One thing that we were interested in particular, right, is that we've seen how important these different microbe-microbe interactions appear to be in shaping dynamics. So what does happen if you change how microbes are interacting with one another? And so, do it for 1 million communities, as Tom so nicely said, right? We can do hundreds, thousands, millions of simulations. And so, for example, you can say, well, what happens if we systematically change how microbes are interacting with one another from, say, fiercely competing for nutrients to space to lovely cooperative communities where everyone is cross-feeding and everyone's helping one another out, weak interactions to strong interactions. And for example, one thing we see, is that these sort of positive interactions, so things like these cross-feeding sort of relationships, are really important for driving predictable microbiome assembly. And this is in hindsight very obvious, right? If taxa rely on one another to grow, then of course, in order for me to colonize, I need my partner to be there, and it sets up this ordering. But what we also see, and this is actually a much older result, is that the more taxa interact in a positive way, actually the less stable a community we would expect, we can discuss, but we would expect the less stable a community is likely to be in the face of antibiotic perturbation. So it's less likely to be able to recover if it gets, hit. And so why do I care about this quite kind of wonky, abstract mathematical result? Well, I think it's interesting if you think about the human gut, right, where we see these communities that develop remarkably predictably, yet once they're there, they're actually incredibly stable. So they're really resilient to perturbations. And so it can allow you to think about, well, what do we think is going on here? And we can make a set of predictions, for example. So we think, strong positive interactions between taxa, probably pretty rare. They are just so destabilizing. But they're also important, right? So how do you marry predictability and stability? Well, one idea is that you have positive interactions early as a community is initially assembling, but as the community increases in diversity, you tend to see more competitors, and you tend to see a shift maybe from a more cooperative to a more sort of competitive community. I'm just going to skip through this. The short answer here is that if you look at our infant data, you kind of do see this. So we see that there is this inverse relationship between how helpful a microbe is and how early it colonizes. So helpful guys tend to get in early, nasty guys tend to get in later. And actually, this is a naughty comparison because these are data from different types of experiments. But if we compare the adult gut to the preterm gut, we see actually many more positive interactions within the preterm gut than we do in the adult gut. So yeah, overrunning. So basically, just wanted to touch on this idea, right, that if we want to understand more general ideas of what's shaping community dynamics over time, that actually, we can use they are very simple, right? A lot of assumptions going into this, but we can make some coarse-grained predictions about more generally what we think is shaping microbiome dynamics. With that, I will thank you guys. The baby project started while I was visiting Seth's lab in Boston, various other people who are also really wonderful, most importantly, Chitong over here. The final thing I will say is that I am recruiting. I do not yet have a picture of my lab, so instead this is a lovely picture of the Peak District. You can come and you can visit the Peak District. And so yeah, do get in touch if you're interested. Yeah. So the short answer is I don't know. This guy down here is a PhD student of mine, and he's really trying to figure this out at the moment. So we now know some things. So you can do aerobic and anaerobic. That doesn't change things, so it's probably not that. I was really hung up for an idea for a while that it would be ion limitation, because particularly in preterm infants, iron's really important. And if you think about plus-minus interactions, well, siderophore scavenging seems a really appealing idea, but he's done some nice experiments, and it doesn't, we can limit iron in the media, and that at least doesn't change what's going on. The most recent little piece of data we have is that if it's a bit grim, but you can take mice, you can sacrifice them, chop out their guts, and then basically wash through the contents, filter that, so you've got no other microbes, but you've got like a gutty mush kind of thing. And there it looks like, again, we're seeing, I hesitate to say for definite, but in our preliminary data, it looks like maybe we're seeing that plus-minus interaction come back, which maybe suggests it's at least not a very active host immune response, because you then don't have, yeah. Yeah, so this is where I think if it's robust, and I really want to hesitate, but if it's robust, this like gut wash thing, we think probably that's going to have killed all of the macrophages are going to be all mushed up and stuff. So it would probably not be that, but I don't know, basically. We were trying to figure it out. So, yeah.
Yes. Yeah, thank you for that. I was wondering with the way that some there's parts of my mind that
Yes, definitely. Yeah, so it's stool, right? So we probably have a pretty good picture of the like lumen of the gut, but what we know we will be missing is those guys that really sit in the mucousy epithelial layer, and what we hope is that probably while they are undersampled, there is a consistency in the level of undersampling. So if you see these sort of mucousy guys increase in your stool sample, that probably corresponds to an increase in the mucus, but we're certainly not getting as much as we would like.
So, under very general conditions, if the intraspecific terms of the locable equation are negative, you get stability. Have you tried imposing negative intraspecific interactions?
In the fitting or in the theory?
In the theory, it is none. Like
Yeah.
Intraspecific interactions, and then you don't have any interspecific interaction.
But you mean in fitting the yeah. So
negative terms in the diagonal.
Yeah, so I've tried that. You end up with really I was speaking to someone else about this. The more so we use a Bayesian approach, and the more of these assumptions you put in, the more gnarly it gets to get your models to converge. But the short answer is actually we tried this, saying okay, the intra-specific interactions have to be negative. You don't see any major changes to the sign of the interactions that you're inferring. In fact, most of the time you're inferring negative intra-species interactions anyway. Yeah. Cool. Yeah, sorry, I overran.
You can wait here. Jamie, can you set up? Okay, so now you can answer. I just wondering if there's a difference between registered infants and
So in full-term infants, yes, this is a known thing that you get at least subtle differences in microbiome composition breastfed versus formula-fed. I shouldn't have unplugged my thing actually, because I have a slide on this. In our preterm infants, we don't see any obvious differences. It's a funny thing, we don't see obvious differences in formula fed versus breastfed, and we see basically no difference in terms of are you vaginal or C-section birth. And I think probably what's going on here is this fact that preterm infants are a pretty weird cohort, right? They're, you know, these poor kids, some of them, they're so tiny, right? And there's so much else going on. And they're often alternating, often like one day they're going to be breastfed, the next day they're on formula, the next day they're nil by mouth. And so I think probably it all shakes out in the wash, those things that really do matter for a full-term infant. Yeah.
I have a technical question. So when you do the logical terrafilling, you need to get the rate. But the rate, given the sampling frequency, the rate can be noisy.
Yeah.
Right? And how do you deal with that problem?
Yeah, so we deal with it so in this way of we end up with a fixed point estimate with some like a sort of standard deviation of confidence around that fixed point, but we are assuming that basically, again, things shake out in the wash and that, you know, we have an average estimate of the growth rate plus some. Did that really answer your question? Possibly not.
Possibly.
No,
Possibly.
maybe rephrase your question.
We can then talk later. Yeah.
Yeah.
Can you the screen? Yeah. Okay. So, let's see. Thank you.
Oh, yeah.
Thank you.
I'd run away with it, you know.
Great. Our next speaker is Jamie Lee from Imperial College London.
Wait, sorry.
Hi, thank you for this opportunity to present my work today. My name is